to prepare the agarose gel. Agarose is dissolved in trace acetic acid buffer, pH 8, with heating in microwave until agarose is completely dissolved. During electrophoresis, pH 8 should be kept because at this pH the DNA fragments are negatively charged and this allows them to move towards the positive electrode. Before loading the gel casting tray is prepared. The tray has a bottom and 2 walls. The other 2 walls are movable. They should fit tightly to the tray. Appropriate number of combs is placed in the gel tray to form the start wells. The gel plate should be taped securely and contain the wall combs prior to pouring. Ethidium bromide is added to the cooled gel. Ethidium bromide allows visualization of the result after the electrophoresis. It is very toxic, therefore the tip should be thrown into the container of hazardous waste. The solution is gently mixed and left for a time until no bubbles are released. Then it is carefully poured into the gel tray and left to solidify. While gel is solidifying, the DNA samples are prepared for electrophoresis. Bromphenol blue, dissolved in glycerol solution, is loaded into the wells on a special plate. Then the sample of the DNA amplified are added. Bromphenol blue is an indicator, which becomes blue due to increase of pH in presence of DNA solution. When the gel has solidified, the comb is carefully removed from the gel. The start wells should be well formed. The gel plate is placed into a submerged gel electrophoresis apparatus. The DNA samples are carefully loaded onto the wells in the gel with Eppendorf pipette. The tips should not touch the gel. The samples should be dropped above the well. The DNA samples are heavier than the solution and fall down due to the presence of glycerol. When all samples are loaded onto wells in the agarose gel, the apparatus is closed and the electric field is switched on. The charged molecules start moving in the agarose gel towards the anode, the positive electrode, because they are negatively charged. Depending on the molecular mass, they reach a definite position in the gel. The DNA bands are visualized using ultraviolet light box. Protective glasses should be used. The DNA fragments in each sample have reached one and the same position since one and the same fragment from DNA has been amplified. The molecular mass of the DNA fragments is determined using the standards in the first well. The standards are a mixture of DNA fragments with known molecular mass, i.e. with different length. The presence of amplified fragment in each sample shows that the polymerase chain reaction has been successful.